everybody. My name is Rebecca DeJesus. I am a junior here at the University of Texas at El Paso, and welcome to the Border Biomedical Research Center. Here at UTEP, I work in Dr. Renato Aguilera's lab doing cancer biology. My job is to do drug discovery, to see if new drugs are cytotoxic to different cell lines, and the biggest part of that is working in cell culture. Here at UTEP, the portion of my work is in cell culture, where we pride ourselves on aseptic technique and cleanliness. We have over 200 different cell lines in which I work on 10 different cancer cell lines, each of which have different morphology, cell complexity, and therefore have different media and culturing types. An example of this is the OV90 line from ATCC, which I had the pleasure of unfreezing from ATCC, creating its media, and freezing them for future use. I'm also the only person to experiment on this cell line. Cell culture is very important to my work here at UTEP and helps us differentiate our results for every experiment. Here at the BBRC, we do a differential nuclear staining assay, or DNS. This is what we use to determine cytotoxicity of various compounds. Here we stain cells with two different dyes, blue being live called Hooke's dye, and red being dead called propydium iodide. Then we incubate and we take pictures of them at said incubations. Based on the fluorescence we get, we can determine the CC50, or the cytotoxicity concentration at which 50% of the cells are dead or inhibited. The flow cytometer is used for many different assays, two of which are annexin and JC1. The flow cytometer detects cell complexity and fluorescence and shows cell population of those two. Annexin is used to determine apoptosis to see if our compounds induce apoptosis on the cells, while JC1 determines if an intrinsic pathway, being mitochondrial depolarization, is used and is showing monomers versus aggregates. Both of them are used through flow cytometry to determine apoptosis and intrinsic pathway analysis. One of the best experiences that I have had being a part of RISE is going to ABRCAMS, the annual biomedical research conference for minority students, where I got to present my work on cancer biology and the newfound drugs that we have found. I could not have done it with the help of many mentors, being Dr. Armando Varela, Denise Gutierrez, and of course Dr. Renato Aguilera, along with Ms. Gladys Almodovar. And everything that I have shown you today is on the poster, from DNS to annexin to JC1. Everything is here, and this is my entire project and what I have been working on since I have started here at UTEP and in the RISE program. So being a part of RISE and doing all of this cancer research has made me fall in love with research, and I want to do it for the rest of my life. I want to go to grad school and pursue a PhD in biology or in cancer biology, to then further my research that I have fallen so much in love with. Thank you so much for watching.